Severe acute respiratory syndrome, or SARS, is a serious form of pneumonia caused by a virus called SARS-associated coronavirus, SARS-CoV. SARS may sound familiar because there was actually a huge SARS outbreak in 2003. According to the World Health Organization, a total of 8,100 people worldwide became sick with SARS during the 2003 outbreak. Of these, just under 800 of them died. In the United States, only 8 people actually had laboratory evidence of having a SARS-CoV infection. All of the people affected had traveled to other parts of the world where SARS was quickly spreading. By electron microscopy, researchers have identified the morphology of SARS-associated coronavirus as a pleomorphic, enveloped virus between 60 to 120 nanometers in diameter. Viral particles are spherical and form large clusters. Lastly, there are cilia-like protrusions outside the envelope. They usually line the surface of the plasma membrane. SARS is a zoonotic agent, and its reservoir is rhinophid bats. Its host range is humans, cats, pigs, raccoon dogs, Himalayan palm civets, and Chinese ferret badgers. The outbreak in 2003 was caused by a virus being spread from these bats to other animals, most likely civets, and then finally infected humans in southern China in 2002. The incubation period of the virus is usually 2 to 7 days, although in some cases it has been as long as 10 days. SARS spreads mostly by direct transmission, or close person-to-person contact. The virus transmits directly when respiratory droplets, produced when someone coughs or sneezes, come into contact with the mucous membranes of uninfected individuals, such as their eyes, mouth, and nose. The virus can also be spread by indirect transmission when uninfected individuals come into contact with contaminated objects, fomites, that had previous contact with infected people. Additionally, the virus can be spread by airborne, fecal-oral, and fecal-respiratory transmission. The greatest risk of transmission occurs when the viral load reaches a maximum 2 weeks after the viral exposure, increasing respiratory secretions in infected people. When the S protein binds to the SARS-CoV receptor, also known as human angiotensin converting enzyme 2, ACE2, the virus envelope fuses with the cell membrane. Next, viral RNA is released into the cytoplasm. The viral genome consists of positive single-stranded RNA, so it can immediately serve as a template for translation to make viral proteins. Viral RNA synthesis takes place in membrane-associated replication complexes that contain replicase proteins, replicating the RNA. When the viral genome is replicated, a negative single-stranded RNA is synthesized, and that serves as the template for positive single-stranded RNA. The virus replicates its genome, translates its proteins, and assembles. Virions assemble at the endoplasmic reticulum Golgi intermediate compartment, or the ERGIC. Immature virions move through the Golgi apparatus for maturation, and when mature virions are formed, they are transported in vesicles to the plasma membrane and are released. This replication cycle follows the typical replication cycle of all coronaviruses. The most well-studied and known virulence factor of SARS is ORF6, a 63 amino acid endoplasmic reticulum Golgi apparatus membrane-associated protein. This protein inhibits interferon signaling and blocks import signals of other body proteins. Another virulence factor is the spike protein, known as S protein, which is also a virulence factor in other coronaviruses. This S protein binds to species-specific host cell receptors, triggering a fusion event between the viral envelope and the cell membrane. One of the first signs of SARS is a high fever of over 100.4 degrees Fahrenheit, followed by a dry cough, rash, and diarrhea. Symptoms include headache, body ache, malaise, sore throat, loss of appetite, and trouble breathing or mild respiratory symptoms. An infected person will have trouble breathing as soon as 2 days, but up until 10 days after contact to the virus. There is currently no universal treatment that works for every person infected with SARS. A person diagnosed with SARS should be admitted to the hospital immediately and kept in isolation under close observation. For some patients, they've responded well to antiviral medications such as ritonavir, lopinavir, and ribavirin, and corticosteroids, while for others, this treatment doesn't work. In addition, type 1 interferon, anti-SARS-CoV antibodies, and SARS convalescent plasma have also been used. A ventilator or supplemental oxygen is also prescribed if needed. There is no vaccine or no prophylaxis for SARS. Finally, SARS can be prevented by washing hands frequently, wearing gloves when handling bodily fluids, wearing surgical masks when in the same room as an infected individual, and disinfecting surfaces and personal items that could have been contaminated with the virus. This being said, it is important to take as many preventative measures as possible since there is no treatment or cure for SARS yet.